First of all, thanks very much, Amit and Shona, for this great initiative. It's a great pleasure to be here this evening and to share some of our work with you. And I really want to let you know how much we want to support the Glasgow University Psychiatry Interest Group. It's a great thing to be part of, and it's a great pleasure to be here this evening. So as Amit touched on, I work on the interface between immunology and neuroscience. And the idea behind that is really to push forward some developments within biological psychiatry, leading to, we hope, new treatments. And I don't think I have to persuade anyone that really new treatments in psychiatry are paramount interest, really. We have some of the most disabling disorders in medicine, dementia, depression, the psychoses, many of which, in fact, all of which have been devoid of any new treatments in the last 20 to 30 years, really novel, transformative treatments. And to give you some examples, in rheumatology, for example, when I was a medical student, people died of rheumatoid arthritis in their 50s. That doesn't happen now. Similarly with MS and neurology and Crohn's disease and gastroenterology, all of which have benefited really from massive paradigmatic shifts in treatments, much of which has been driven by a greater understanding of the role of immune-mediated inflammation. And to bring it back to psychiatry, most practicing psychiatrists will recognize these symptoms. This is a group of symptoms we call sickness and behavior. But actually, most of the people we see will experience fatigue, low mood, impaired cognition, as well as the more dramatic symptoms of the psychoses. And we've always struggled to really understand how these things fit together and what drives them from a neurobiological perspective, never mind trying to get rational treatments. So what I'm trying to do today, I think, is to try and share with you what we are trying to do from that perspective. And the key thing at the bottom of this is the impact of inflammation on the brain itself is the key driver. And so we have a variety of different studies in our group here in Glasgow, some of which are essentially human studies, experimental medicine studies, and a study here that's developing new drugs for depression and dementia, which I'll share with you at the end. And the others are more mechanistic mouse-based studies. Essentially, these are studies that are trying to understand the mechanisms that we couldn't really drive forward in humans for a whole variety of very understandable reasons. And in terms of those mechanistic studies, we use a lot of mouse models, and some of them involve really stimulating the mouse in the periphery. So, for example, this is a skin-based model, and you can cause a systemic inflammation in the model, not too much detriment to the mouse, really. And in essence, what you're going to do then is engender an immune response in the brain itself. And it's that response within the brain that we really want to target and drill into and find out what the consequences of that is. And we do this in a whole variety of different ways, using classical histology and staining, where we can look at neurons and their response to that level of inflammation. We can look at that from the point of view of how that fits together in terms of the structure of the brain. And then we can take that a bit further forward and look at how that affects brain connectivity. And you might hear a lot about this in terms of brain imaging nowadays. Neuroimaging tends to talk about connectivity, whether it's functional connectivity or effective connectivity, and neural networks rather than classical neuroanatomy. And you can do this in very cool ways, really, by using a whole variety of different so-called optogenetic techniques. And those allow you to look at how one neuron will speak to another, and you get a classical neurophysiological readout on the basis of that. So you can actually do this both in vivo and ex vivo. And in fact, the person who developed optogenetics was a psychiatrist called Carl Dicerot in Stanford. And some people were saying that he might actually be a candidate for the Nobel Prize. This is another way that we can do this. We can use live brain slices. We can take the mouse brain and create so-called organotypic brain slices. And these are slices that allow you to look and see how the neurons themselves operate in real space, as it were, and how they relate importantly to other cells in the brain. And why is this important? Well, we have to relate all of it to behavior, because ultimately, as psychiatrists, we're interested in the consequences of these biological changes and how they drive the behaviors that we think are related to the phenotypes we see in the clinic. And we can use all sorts of different techniques. We can look at how the mouse relates to mood, if you like, and so-called anhedonia in mice. So mice don't get depressed but they certainly lose interest in things, and they lose the desire to satiate pleasure, if you like. So we can measure all sorts of things using different behavioral methods. And bringing that back to the human, one of the great struggles we had before was trying to measure inflammation in the human brain in vivo. And those techniques have now advanced enormously, actually, and we can use from previously using very high radiation heavy techniques to radiation low techniques like MRI. And you can look at the way that essentially water molecules are moved by inflammatory molecules, and that gives you an idea of the impact of inflammation in the brain in real time and obviously in vivo. And one of the studies I mentioned there is also trying to understand not only how that works in vivo in humans, but also how it works in terms of brain chemistry. So we can do that again using MR techniques. This is magnetic resonance spectroscopy this time. And that allows us to look at things like glutamate. And glutamate, you'll all be familiar with, is the excitatory neurotransmitter and actually the subject of many drug developments as we speak. And our idea there is that actually what you've got in inflammation, amongst other things, is a significant increase in extrasynaptic glutamate, and that is damaging to neurons. So we think that impacts on the connectivity that I talked about earlier on. And in terms of actual treatments, so we've one of our welcome studies has now led us to the development of 2 new drugs, one for depression and one for dementia. And this is the depression one, which is actually targeting a key receptor in microglia. These are the innate immune cells of the brain, and they produce lots of these cytokines which we think might be damaging. And if you block one key receptor on those, then it stops the production of those. And so that's the thinking behind this new drug, which we're, and Nagori Panades is also involved in this study. We're about to start that trial anytime now, actually. So that's a trial for a drug for people who've got treatment resistance, and that constitutes about 30% of depressed people. And then lastly, of relevance currently is the impact of COVID itself, which is, of course, in some ways, rather tragically, a classical experiment in what the impact of inflammation and virally driven inflammation is on the brain. And so we're part of this UK-wide study funded by the MRC called COVID CNS, where we're looking at the acute neurological and psychiatric complications of COVID-19. And we're trying to work out what the mechanisms might be, because this is it's a fiendishly difficult virus to study, because it doesn't actually get into neurons, even though it causes quite substantial neurological complications. So we're trying to work out why it causes that without actually infecting neurons themselves. So I'm going to stop there, because I think that's probably me chuntering on it for long enough, and hand over